Your Unit 3 notes for genetics. We're going to start by talking about DNA mutations. So a mutation is a mistake in the DNA of an organism. That mutation is going to alter the genetic information, and as a result, it can affect whatever protein is being coded for. So gene mutations affect specific parts of the DNA code, and there are a couple different ways that the gene can be affected. So there's two main types of gene mutations. There's point mutations and frame shift mutations. Point mutations are changes in just one nucleotide. So basically one letter of DNA is going to change. As a result, it's only going to affect one amino acid, right? One letter is just going to be switched for a different letter. This is also sometimes called a substitution mutation. That's when one nucleotide is switched for another. So if the DNA has an A, the mutation might change one of the A's to a C. It may or may not change the amino acid depending on the letter it changes to, because multiple codons code for the same amino acid. It could have no effect at all. Then there are also frame shift mutations. Frame shift mutations are when a nucleotide is added or deleted from the DNA. As a result, when you add or delete a nucleotide, it's going to shift the rest of the DNA strand. So if you add one, the whole thing is going to shift over to make room for that new nucleotide. If one is deleted, then it's going to shift to close that gap. So all of the amino acids after that mutation are going to be affected when the DNA strand shifts. As a result, the protein typically cannot carry out its normal function. That protein is not going to be usable. So here are some examples of what we were talking about. So if this is our original sequence, a point mutation is just going to change one nucleotide. So you can see this T, the mutation just changed it to a C. The rest of the nucleotides are all in the correct spot. For a frame shift mutation, if it's an insertion, meaning we're adding a nucleotide, then all of those other nucleotides are going to shift over, right? If we take one out, they're going to shift the other direction. So you can see the result is very different from our original sequence that we have over here, right? So it's going to affect all of the codons after that mutation. So let's talk about meiosis. So let's talk about the chromosomes organisms have. We've talked about this a little bit before. Body cells, which are also called somatic cells of most organisms, have two sets of inherited genes, one set from each parent. So you have one set of chromosomes or genes from your mom and one set from your dad. As a result, each body cell has two genes for each trait, right? One gene from mom, one gene from dad. Sex cells, also called gametes, which are eggs and sperm, they only have one set of genes. So those two gametes are going to combine in order to create a new organism. That's what those sex cells are used for. So eggs and sperm have half the number of chromosomes of a regular organism. That way, when they combine together, we can create a new organism with the correct amount of chromosomes. So let's look at this in terms of numbers. Human body cells have 46 chromosomes, 23 matching pairs. So we get 23 chromosomes from mom and 23 chromosomes from dad. So 46 total. Human gametes, so human eggs and sperm, only have 23 chromosomes. So that when that gamete, that sex cell, combines with another one, you end up with that correct number of 46, right? So eggs and sperm only have 23 chromosomes in humans. A human egg and sperm fertilize to form what we call a zygote. A zygote is the first cell of an organism that has all 46 of those chromosomes, that has the correct number of chromosomes. Eventually, that zygote is what's going to develop into a new human. So what is meiosis? Meiosis is that process in which the number of chromosomes is cut in half in order to create those sex cells needed for reproduction. So because eggs and sperm need to have half the number of chromosomes, meiosis is the process that's going to allow that to happen. During meiosis, we're going to recombine the genes from an organism in a unique way so that the eggs and sperm that are created are going to be unique. Every single one is going to be a little bit different. So here are the steps of meiosis. First thing that's going to happen is that the DNA is going to replicate. So if you look in this diagram, a human has 46 chromosomes. the DNA is going to replicate so that we have 92 chromosomes. Then there's going to be the first division. After the first division, we have 2 cells that have 46 chromosomes each. Then each of those cells is going to undergo a second division so that it's cut in half again, right? And then the result is 4 gametes, each of them with 23 chromosomes. Right? So we start with 46, the full number, double that through DNA replication, split it in half once, then each of those is going to split in half again. So 2 divisions resulting in 4 gametes. So 4 cells that have half the number of chromosomes. Crossing over is something unique to meiosis. It is going to happen before the first division. So before it divides for the first time, crossing over is going to happen. What happens during crossing over is that those chromosomes are going to match up. So your copy that you have from mom and your copy that you have from dad are going to match up with each other. And they're going to swap information between the two. As a result, the chromosomes you are left with have a mixture of DNA from both parents. So if you look at this diagram here, this red one may represent mom, the yellow one may represent dad. When they do crossing over, they're going to swap pieces of information. So as a result, now we have 4 unique chromosomes, right? This one is fully red. This one is partially red, partially yellow. This one is a little more yellow, a little bit of red, and then all yellow. So 4 unique combinations. So meiosis is going to divide the number of chromosomes in half. That's the purpose of meiosis. If we didn't have meiosis, each generation would just have more and more chromosomes, right? If our eggs and sperm had 46 chromosomes, that means your child would have 92 chromosomes. And then their child would have 184 chromosomes, and so on and so forth. So we would just have a huge number of chromosomes after even just a couple of generations. So with meiosis, cutting that number of chromosomes in half makes sure that every new generation has the same number of chromosomes as their parents. The purpose of crossing over is to ensure that each of those gametes, each sex cell, has a unique combination of DNA. This is why 2 parents can have many children that are unique, right? Like I have a sister and we have some traits in common, right? Like we both have blue eyes, but we have different colored hair, right? That's why 2 parents could have 10, 15 children that are all going to be unique from each other. It's because of crossing over. Make sure that each sperm and egg has a unique combination of genes. All right, and then into genetics. So what is a gene exactly? A gene is a section of a chromosome that codes for a trait. Alleles, that term allele, means different forms of the same gene. We represent those alleles with a letter. For example, the trait may be eye color. The alleles are going to be different forms of that gene, right? So B, the capital B, could represent brown-eyed allele. A little b could represent the blue-eyed allele, right? So different alleles represented by a letter. The overall trait for this example would be eye color. Genes can have dominant or recessive alleles. So dominant alleles are alleles that are expressed or shown when different alleles are present. So we represent dominant alleles with a capital letter. For example, a capital T may represent a tall plant and a capital P may represent purple flowers. If the organism has that allele, has that capital letter, that's the trait that's going to be shown. Recessive alleles are not shown or not expressed when the dominant allele is present. They're represented with a lowercase letter that's the same letter as the dominant allele. For example, a lowercase t would be a short plant. Lowercase p would be white flowers. So you are only going to see the white flowers if there's no capital P, right? So you have to have 2 of the lowercase letters to see that trait. What is a genotype? A genotype represents, or a genotype describes the genetic makeup of an organism. So it is going to include both genes that a person carries, one from each parent, and it's written in the form of letters that represent the genes. For example, big B, little b. That's what we call it, right? Like capital B, lowercase b. Big B, little b would be brown eyes. Their genotype is big B, little b, right? That's describes the actual genes the person carries. Or big P, that would represent the actual genes the person carries, one letter representing each parent. There's a couple different terms we have to describe genotypes. So homozygous genotypes have two of the same allele. So it's either going to have two dominant or two recessive. For example, big B, right? Two of the dominant letter, or little s, two of the small letter. So that would be big B would be homozygous dominant. Little s would be homozygous recessive. Heterozygous genotypes have one of each allele. So it's going to have one dominant and one recessive, which is going to look like one capital and one lowercase. For example, big R, or big Y, little y. Both of those would be heterozygous. So what is a phenotype? The phenotype is the physical expression of the trait. We write that as words. It's the words that describe the trait the person has. For example, green eyes. That is the trait that we see. It's how we describe the trait the person has. Or a button nose, or green peas, right? All of those are the phenotype. The phenotype is the physical trait that we actually see. You can't see the letters or the alleles that the person has. That would be their genotype. The phenotype is what we physically see that we can describe with words. Right, so here are some examples. So the homozygous dominant genotype would be big T. The phenotype would be tall, right? It has that dominant phenotype. Homozygous recessive would be little t. The phenotype would be short. It shows the recessive phenotype. The heterozygous genotype would be big T, little t. The phenotype we would see is going to be tall, right? Because that dominant is going to overtake the recessive. And we would call that a hybrid, right? Has one of each. So we call that hybrid. So what is a Punnett square? Punnett squares are used to predict possible offspring genotypes based on their parent genotypes. When studying inheritance of only one trait, this is called a monohybrid cross. So with a Punnett square, we can take what genotypes the parent has and predict the possible offspring, right? We can determine like the percent chance of the offspring having certain traits. So here's how you do a monohybrid cross. First thing you have to do is determine the parent genotypes. Remember, those are the letters. Then you're going to separate the alleles. So on one parent will be at the top and one parent will be at the side. You're going to separate those alleles so each one has its own row or column. Then you're going to combine those parent alleles inside the boxes. And remember that the boxes represent possible genotypes of offspring, not the actual offspring, right? This is just used to predict what we would expect to see, right? These are possible genotypes. So this is how you would set it up. So we have our two parents here. One parent is big Y, little y. So we have we separate those, one on top of each column. The other parent is also big Y, little y. So we separate those, one for each row. Right, so like these on the side might represent mom and the top might represent dad. Right, we would show this as like a genetics problem as big Y times big Y, little y. In this particular example, big Y is representing the trait for yellow, little y is representing the trait for green. So when we complete it, you follow this big Y down. So both of these are going to have a big Y from the top. This little y goes down, this big Y across, that little y across. Whenever we write our genotypes, we always put the capital letter first, right? So even though you might move the little letter first up, across, or down, we always are going to write the capital letter first. So once we have our Punnett square filled out, then we can determine ratios and percentages to help us describe what we would predict those offspring to look like. Percentages for a monohybrid cross, when we only have 4 boxes, are calculated with each box representing 25%. So if I was going to look at the genotype percentages of this, I'm going to count how many boxes have each genotype. I have one box that's big A, so that would be 25% big A, big A. Two boxes have big A, little a, so each of those is 25%, so 50% total, 50% big A, little a. And I have one box that's little a, so 25% little a, little a. So if, let's look at the trait for this, the big A is going to represent a straight pinky, little a is going to represent a bent pinky. We can also determine ratios and percentages for the phenotypes. So for the dominant phenotype, how many boxes are going to have a straight pinky? It's going to be 3. So all 3 of these boxes are going to show straight pinky because that big A is always going to show even when it's with a little a. So that would be 3 boxes, 25% each, so 75% with a straight pinky. Then we have 1 box with 2 little letters, so 25% with a bent pinky. That's how you do percentages. So ratios are going to represent just the number of boxes with each genotype or phenotype. So we're just counting the number of boxes for a ratio. For example, for the genotype, I'm going to count the number of boxes with the dominant homozygous, number of boxes with the heterozygous, number of boxes with homozygous recessive. So I have 1 box with big A, 2 boxes with big A, and 1 box with little a. So my ratio is going to be 1 to 2 to 1. So when you write a ratio, you always put a colon between each number, and we read it as 1 to 2 to 1. For my phenotype ratio, I'm going to count the number with the dominant trait and the number with the recessive trait. So I have 3 with the dominant trait, 1 with the recessive trait. So my ratio is going to be 3 to 1. All right, those are the end of your genetics notes. Make sure that you complete the entire document and turn it in on Canvas.